Hi, I'm Luzark, and also Richards is here. He'll be helping me out a little bit too today. Excuse me. As Eric said, we're going to be discussing guidelines for the identification of Baetis adonis and Baetis tricaudatus in Southern California samples. Tricaudatus have in the SMC and other swamp samples in Southern California have given us the most problems in our data sets as far as identification. And what we want to lay out here is a set of guidelines for you guys to do so that we can everybody on the same page for dealing with these difficult taxa. The problem with these taxa is some recent studies initiated by SQURP looking at the DNA of a lot of our bioassessment critters. One of the results of that study turned that it appears that Baetis adonis and Baetis tricaudatus are not good species, but are probably species complexes. And to give a little bit more detail and background of this problem, I'm going to ask Jeff Webb of Rithron to come in now and talk about this a little. Jeff is a mayfly specialist, and he's been working with ABL and with SQURP on this project, and he's got a much better handle on what's going on here, and he can explain a little about the species complex here. Sean,
Can you hear me all right now?
Yeah.
All right, good. So thanks, Joe. I think you've hit it on the head by saying that, yes, adonis and tricaudatus are both species complexes. As part of the work with SQURP, when I was at the University of Guelph, we sequenced the standard DNA barcoding gene, cytochrome oxidase 1, for almost 900 specimens or something, but it's part of the SQURP project. Those two taxa, as well as tricaudatus from all over North America. And the tricaudatus is a mess. We have a hugely variable species morphologically, and it has a lot of synonyms, and it's basically become default identification almost for a lot of the Rodani complex Baetis with the robust CD on the ND and on the paraprox and such. And adonis has never been a really well-known species. And when we looked at the California stuff with the barcoding, that I don't have the data here with me, but I believe that we had two distinct clusters of tricaudatus and two clusters of adonis. And what was really interesting is that I think it came out with adonis being the sister to one of the tricaudatus, and then the other tricaudatus being most closely related to the other adonis. It's very well separated genetically either. Each of the two sister pairs, they only had about 1% genetic differentiation, and usually we find species with species you see less than 2% variation, and between species grade 10. So we had these 4 clusters that on each of the 2 branches had 2 taxa. Make any sense?
Yes.
Yeah. Anyways, so not terribly well separated between The two adonis were well separated, the two tricaudatus were well separated, but one of the adonis was closely related to the tricaudatus, and same thing with the other adonis and tricaudatus. And the molecular data, using the barcoding data in general, seems to work really well for separating the species, but maybe Joe will talk about this later, that it seems there are some good ecological characters and potentially morphological characters for separating these things. So is the type of data people does that sound good?
Yeah, that gives us a little bit of background here.
Oh, hold on. Mute here.
The fact that right now we kind of know that these species are probably at least 4 species. Brady and I have carefully looked at a lot of the characters, and we can't really come up with anything else right now that we can give you to sort these suckers out. And what we're going to do is we're going to propose something, basically a set of guidelines for what we want folks to do with their specimens and their samples. Now, until somebody can figure out better what to do till some of us can figure out how to identify these guys using morphological characters more accurate. So, Brady, I don't know if you want to come over and talk to Brady. After we've basically sat and looked at literally live mounted thousands of specimens down there, and we can't really come up with a good way to separate out all of the immatures. And the larger specimens, the only character that we can find to at least separate these critters out to a nominal species name that's actually in the literature is in the gill character. So kind of what we're gonna propose is that, first of all, we would like folks to only attempt to ID fully developed Baetis larvae to species in Southern California, and ID all their other Baetis larvae just to genus. And again, the only character we found to distinguish between Baetis adonis and Baetis tricaudatus is the spawn character on the gills. And if the gills are missing, we'd like you to ID the mature larvae also as Baetis. The reason why we're doing this is basically so that in this region we try to have more consistency in the data reporting. Until further study is done on potential morphological characters to separate it, and there are some possible ecological characters, although I'm not familiar with them, but I mentioned them briefly, that we may be able to use in the future. I think this requires a lot more study. One of the reasons why we would like people to try to ID the mature intact Baetis adonis tricaudatus in Southern California is so we can get a pool of specimens from that region of mature larvae, so that maybe we can use those in combination with a more in-depth molecular study in the future, help sort out what's going on down there. Brady, do you have anything else to add about
Well, to clarify, when we say the specimen pool, what we really mean is you set those specimens aside for reference. Keep them available so that we can get somebody who knows the stuff really well, who's going to study them, Jeff Webb, or someone else. Have a nice pool of really good intact specimens so that they can look at them. Everybody on this call now, basically, who's done the Southern California work has had about 119 Baetis or some 300 Baetis that they have to go through and look at for these spines. And I know that both have taken the work here in the lab or the external QC work that I do and slide mounted every single one of those. So it only requires a lot of work, but we think if we create a specimen pool here, it'll get good specimens for whoever's going to work on this.
Okay. So to continue on with the presentation, the next couple of things I'm going to do is I'm just going to put up a couple of slides on how to tell the difference, the cutoff point between what we want as mature larvae and everything else that we would consider immature larvae. And I'm going to show a couple of slides of the characters to use on the adults, and then I'm going to go into I have some specimens here from Southern California, and I'm just going to go through the specimens and kind of show what the mature larvae look like that we'd like you to attempt, and basically show you what the specimens that we just want you to leave at Baetis from now on look like. And after that's over, we'll be going on to maybe to do some more questions, and we could talk about this maybe a little bit. On the next slide. If you don't know how to identify fully mature and fully developed Baetis larvae, these are two photos of specimens that I just took the other day from some Baetis from San Diego. You can see on this specimen here, this is a fully developed larva. You can see how the wing pads are very, very large in relationship to the body, and if you look here carefully, you can actually see the developing wing inside. And over here is a larvae that's actually almost completely is pretty much ready to go. In fact, you can see the exoskeleton right here is just starting to split. You can see inside the wing pads, they're dark like this because the entire forewing is completely developed and it's folded up inside now. And actually, this is a male. You can actually see right here, eyes, and then on top here you can see the developing male, the eye of the male. So these are the specimens that we would like you to separate out from your other Baetids and to try to ID. Let's move on to the next slide here. Immature larvae. If you look at these guys have like much smaller developed wing pads. And even in this wing pad, you can see there's some striations in here in this wing pad for the developing wing. But this is still, we consider this still an immature larva, and we would like you guys, anything like, like from this big, smaller, to try. Basically, unless it looks like guys, we prefer that from now on that you leave specimens at this. To determine the difference in mature larvae between putting the name adonis and tricaudatus on your specimens, I feel there's really only one reliable character, and that is the large spines on the margin of the gill, and this will require that you either remove the gill from the specimen and make a mount and look at it underneath the compound microscope, or you can the whole specimen, but it doesn't work very well a lot of the time, and just look at the margin. But they're much easier to see under the compound microscope, and even though it sounds like an onerous step to pull the gill and wet mount it, I found that it's actually saves you a lot more time than trying to like manipulate your specimen and manipulate your light to see the shine of the spines. They will underneath your dissecting scope. You can potentially see them underneath the dissecting scope on high power. If you pull the light just right, the spines will catch the light and shine. But I found that it's much quicker to pull a gill. But I do tricaudatus. They're just going to have this serrate gill, and it's basically the trick here is like you want to look at. You're looking for adonis, and if you see these large spines, you're going to name adonis on it. Otherwise, you'll end up putting the name tricaudatus on them. I can actually, let me just, see this? See that? Oh, you can't see it. Okay.
No.
That's not up.
What you want to do is share your desktop, I think.
Okay. All right. I can, when I go to this in later on, I can show you like a larger view of these from a point slide. Actually, that's it for now. I'd like to go into the live stuff. Eric, if you could switch me over to be the presenter. Yeah?
Do a quick start tab and then your desktop.
Okay. Quick start. All right. Thanks. Actually, hold on a second. Can you see this now? This big blow-up picture of the bonus gill. Can you see that? Yeah, all right, it's on yours. You can see a better view here, and I blew it up in Photoshop here of the spines. I just show folks that real quick before we go into the specimens. Good? Okay. Okay. Specimens. These are all Baetis that were collected down in San Diego County, and you've got I have a very mature Baetis here. And these are the kind of guys that you'll want to take and pull the gills off of. What? Oh, okay, sorry. Sorry, folks. I'm working on the scope while I'm doing this. I'm not looking at the screen. So you'll want to pull the gill off of these guys. wide mouth, look for lines. This example of mature larvae that doesn't have darkened wing pads. Again, you can see the striations in the wing pads. A little bit more light here. And we'll develop larvae. And in the past, you can see the color patterning here. In the past, I would have called that adonis right off the bat, but doesn't anymore. Again, there's other mature larvae. Mature larvae. Now we'll get some immatures here. This is something that we would want you to leave at Baetis from now on. This. Like that. Hold it. Okay. Thank you. Ray told me that these aren't showing up. So like that. We would want you to leave at Baetis. Let's see. I would even say that something like this, you should leave at Baetis. Just leave it at. And the obvious answer is if it's mature and it doesn't have gills, you have to leave it at Baetis too. Okay, I think that's all we got here. So
to take some questions at this time?
Yeah, I think this would be a good time if anybody's got any questions.
If you have a question, please unmute your phone with *6. Have a conversation with Joe, and then afterwards put us back on mute with *6. Thank you.
Quick question about what keys used to down to Baetis?
Say again?
What keys do you use to identify down to Baetis?
Key I've been using was developed by the Northwest Biological Assessments Work Group's Ephemeroptera Conference. That was in June of 2005, and it was a key put together by Luke Jacobus and Pat Randolph.
Great, thank you.
Okay.
John, and we're a little curious as far as the distribution and kind of co-occurrence of your mature adonis and tricaudatus. How north are you finding the co-occurrence of adonis and tricaudatus?
That's something that Brady and I were actually talking about yesterday, about the geographic distribution of adonis. And we're only seeing adonis in like the LA Basin, San Diego region, find an adonis like in the southern San Joaquin Valley or anything like that. It seems to be very, very restricted. As best we can tell, that they're only down there. We don't really see adonis anywhere in California. Do you want to add to that, Freddie? To the east, we don't work in Arizona ourselves, but I've talked to John Pfeiffer over at EcoAnalyst, and he just told me that he does not see adonis much over there either. Basically, they see Magnus and Nodos over in Arizona. So I'm not sure, but it seems like adonis is very, very restricted in its distribution. And what we've seen and to other people.
Yeah, I would agree here. We see, we encounter Baetis, we see Magnus more than Adonis from the samples in the Southwest. So I was really curious how far you were going up with those Adonis. So thank you, Joe.
Do we have any others?
Wow, looks like I'm getting off easy.
Yet.
Okay.
Hey, it's Jeff at Rithron again.
Hey, Jeff.
Do you guys ever come across Palisadi or Piscatoris at all in Southern California? I know the larvae were only recently sort of described with the illustrations, but I'm not sure if they're in that same area as well. Just more than anything.
No, but a lot of our bioassessment samples are taken at very almost all the bioassessment samples are taken at very specific types of habitat. The kind of wadeable streams, gradient wadeable streams, and they're during index periods, and so we miss a lot of stuff. We know that for a fact. We've done comparisons here where we've done actual surveys of a basin up in High Sierra, and we've done bioassessments and we've done bug surveys simultaneously, and we get twice as many taxa when we do an actual insect survey as opposed to the bioassessment sample. It's the same site,
Yes. I was just curious because there are things that aren't well known, and it'd be good if we could put material together sometime and get some of these problems clarified.
Well, I would really promote amongst all SAFIT folks for collecting mature larvae, and the reason why we want to ID the mature specimens is just basically to hang the name Adonis or tricaudatus on them, and to basically get people to keep them in reference collections with accurate locality data, so that there's a pool of specimens that somebody get their hands on and work on them, work on the taxonomy. For all maybe Palisades and the other one are masquerading down there as one of these guys.
Right.
We're just missing them.
Yeah. Well, they're not well described and not well known. So, thanks, Joe.
Question. This is Rafi, is SAFIT still planning on having an ephemeroptera workshop next year?
Last time I talked to Bob, he said that it sounded like Luke was working on it, and that Luke would let him know by the end of the month whether he would be able to do it in February or not. Bob, if you're on the line, maybe you could on. And
Yeah, I'm here, Joe. That's exactly the status. Luke's going to let me know by the end of the month if it's a go. I think Leon once said he's going to, because he has been working on the manual.
yeah.
And I'm on the line here. Is Jock still there?
Yep, are you still there?
First, I was like, who's Gene?
Yeah, I was just wondering if he was doing a DNA analysis from all over North America, what coming up with Baetis versus tricaudatus?
We did just, I'm no longer doing the DNA stuff, or not at the moment, but we did do some Baetis, and did find them to be distinct from tricaudatus, but not always easy to tell apart morphologically. There are some things called tricaudatus that had very small median caudal filaments. And I believe in Baetis we had two genetic groupings as well, and someone had once worked on this. I don't think they've ever published, but they thought they had morphological differences between these two genetic groups in Baetis as well.
Baetis is a species complex too.
Yeah, basically, the number of synonyms in Baetis, and you've got at least that many things. We've simplified, a lot of these things seem to run together morphologically when you're not looking at really fresh, nice specimens. Now that we started doing this DNA work, I got to see a lot of stuff that was very recently collected, still had good color patterns, and we had adults that were able to associate using the molecular data with the larvae. And the adults, there's very distinct differences in these different things that we've called tricaudatus as larvae. So in some of the larvae as well. Some of the southwestern ones that I've seen have huge, well not huge, but larger gills with really well-developed trachea, whereas most of the others have smaller untracheated gills, and the adult color patterns are quite different. So I think now that I have the additional tool of the molecular data to go with fresh specimens, we can hopefully sort some of these things out, because they do appear to be distinct. When old material, they all just seem to blend together.
Okay,